All right, folks, we have two sections left. We have the cerebrum, the lobes of the cerebral cortex, and then we have the cranial nerves. So let's get going on these. So here you can see the anatomy. You already learned this in lab, so I won't go through all of this. I will point out two very, very important areas. This precentral gyrus right here, that's a primary motor area, and this postcentral gyrus right here is a primary somatosensory. Has really doesn't have anything to do with your special senses, but your somatic senses it has everything to do with. So you can see your lobes, you can see your precentral gyrus and postcentral gyrus, and of course you've already learned those in lab. This is the real brain. A picture of the real brain, not just an artistic drawing, and you can see that there. When you retract, actually when you stick your finger through the lateral fissure, or when you retract the temporal lobes and frontal lobes, you see this insula down in here. That's what you have here is your insula. Your insula does some gustation, sense of taste. It also probably part of the rhinencephalon is in the insula. The rhinencephalon is an area of the brain involved in the sense of smell or olfaction. And the sense of smell or olfaction crosses a couple different lobes. It's partly in the temporal lobe, it's partly in the frontal lobe, and likely part of it's also in the insula. So that's the insula. If you were to stick your finger through the lateral fissure, you would hit the insula. Some people call that the fifth lobe. This is mid-sagittal cut of the brain, and you can see a few things in here. The cingulate gyrus is part of the limbic system. That's the cingulate gyrus right here. We've already talked about the corpus callosum being commissural fibers before. We talked about that. You can see the different lobes from the mid-sagittal cut. You can see the occipital lobe right here, and the occipital lobe, this whole green area. The occipital lobe does sight. It's your visual cortex. The frontal lobe does motor planning. And the very tip of your frontal lobe, the prefrontal cortex, does motivation for your motor planning. Uh, so it gives you motivation to accomplish whatever you're planning to accomplish. So you can see some of that there. The temporal lobe that you can see here does hearing. Call it the auditory cortex. So you can see those cortexes there. Now you can see some different fibers. Three types of fibers. First, we have association fibers. Association fibers stay within a hemisphere, but we have short association fibers, like you can see here, arcuate association fibers. The arcuate fibers go from one gyrus to another gyrus, typically stay within a lobe. They don't cross outside of the lobe. And then we have these fasciculi or longitudinal association fibers, and they go from one gyrus to another gyrus, but they leave the lobe. They're long. So the longitudinal fasciculi are long and the arcuate fibers are short. Then we have fibers that cross from one hemisphere to another, and these are commissural fibers. There's three commissures you learned in lab. You learned the corpus callosum, the biggest of the commissures. You learned the anterior commissure, and you learned the posterior commissure. Then we have these projection fibers. And the projection fibers where they come together is called the internal capsule. You can see that right there. Whoops, I didn't mean to do that. You can see that right there. I meant to underline that for you. So you can see that internal capsule right here. And that's the projection fibers, how they travel out of the brain or into the brain. They could be myelinated fibers going up into the cerebral cortex as well. So those are the projection fibers into or out of the brain. So three types of fibers: association, commissural, and projection. Then we have the basal nuclei. And the basal nuclei do a subconscious level of control of your motor movements. We quite often say that the basal nuclei inhibit unwanted muscle contractions. It's not as simple as that really, but that's one way of starting with it, that the basal nuclei inhibit unwanted muscle contractions. Now think of this. If you have a lesion in your basal nuclei, like Parkinson's disease, and you have the inability to inhibit unwanted muscular contractions, then you would end up with tremors. And that's indeed what happens with some lesions in the basal nuclei is you get tremors because you have the inability to inhibit unwanted muscle contractions. The This right here, the caudate nucleus, the pink thing here, I guess. Yeah, the pink. I guess that's the color. The caudate nucleus kind of follows the lateral ventricles, and it actually probably looks like the lateral ventricles to you a little bit. They hug the lateral ventricles. And if you recall from lab, the lateral ventricles are the two ventricles that kind of look like the Jaguar emblem on a Jaguar car. Here's another cut of it, and you can see the caudate nucleus right here. You can't see the tail of the caudate nucleus. It would be, depending on how, you'd have to cut this a little deeper. It would be down here towards the tail of the lateral ventricle, so it would be down in here. That would be the tail of the caudate nucleus, which you can't actually see here. All right, another cut. Here's another cut coming of the, here we go. This is a coronal or frontal section. And you can see the caudate nucleus right here. You can see the lentiform nucleus here and here. And those are the two big areas of the basal nuclei, the caudate nucleus and the lentiform nucleus. This amygdaloid body is part of your limbic system. It's right at the very tip of the caudate nucleus. The caudate nucleus's tail is right behind that. And let me go back and show you that. Oh, yep, right here. You can see it right here. If you look down here, here's the amygdaloid nucleus right here, and here's the tail of the caudate. So you can see how the amygdaloid's right at the end. And you can see that there. You can see the internal capsule. My projection fibers go down between my basal nuclei. My projection fibers are between my basal nuclei there. These are commissural fibers right here. That's your anterior commissure. This is your corpus callosum right here. So you can see those basal nuclei. All right, now we have the cortical areas. Your frontal lobe is involved in motor movement, and your prefrontal cortex is involved in motivation to do a particular motor movement. It's also involved in predicting possible outcomes of the future, and because of that, your prefrontal cortex is a seat of anxiety. Now think of what anxiety is. Anxiety is stressing about potential future outcomes. They haven't even happened yet, but you're stressing about them. So your prefrontal cortex contains the seat of your anxiety. And in fact, they used to do prefrontal lobotomies to get rid of anxiety disorders. Okay, the planned motor movements occur here in the premotor area, they call it. And then the precentral gyrus is your primary motor area that takes the plan and executes it. So the premotor area is where you put together the plan, and the primary motor area is where you execute the plan, and that's your frontal lobe. Then you have your parietal lobe right here. The big part of your parietal lobe is your postcentral gyrus. It's the primary somatosensory area. And then you have a sensory association area. And for example, you could put your hand in your pocket and you could feel a coin. And right up here in the somatosensory area, you're feeling that coin. You know that it's round and flat and has a ribbed edge to it, and you can sense that. But how do you know it's a coin? The association area associates that feeling with a coin, and that's just from a learning. That's what you learned as you developed from a toddler to an adult. And these association areas contain those associations. The occipital lobe is your visual cortex. Right here at the back is your visual cortex. Then you have your visual association area. And for example, you see something across the field, and then you associate it with what it is. You see an animal across the field, and you say, "Well, that's a deer," and then you even associate that deer with other things. So that's an association area. So the occipital lobe does vision. Your temporal lobe does auditory. Right here's your temporal lobe, and it's the auditory cortex and the auditory association area. You hear a dog bark. How do you know it's a dog? Let's say you can't see the dog. You hear a dog bark. You hear it right here. You associate it with a dog right here. That's the auditory association area. There are diseases where you can hear the dog bark and you can't tell it's a dog. But then when you see the dog, you can say it's a dog. And these diseases are what helped us to determine these different lesions of the brain and what they're doing for us. Then you have this lateral fissure right here. I know it's called lateral sulcus right here. Most books call it a fissure. And you have the insula in here. Now the insula is has your gustatory cortex, your sense of taste. It likely also contains your your rhinencephalon or your olfactory cortex. But let me just tell you that your olfactory cortex is going to be partly in the temporal lobe, it's going to be partly in the frontal lobe, and likely part of the olfactory cortex is also in the insula. But we always say the insula contains your gustatory cortex. So those are the major cortical regions of your brain and what they do. Uh, big area here. This is called the general interpretive area right here. It's also known as Wernicke's area. This is a language interpretation area. This is where you interpret language. And by the way, you have Broca's area right here. This is also it's called the speech center or Broca's area. So when you're hearing someone talk to you and you're responding to their talking to you, there's a fiber going from Wernicke's area directly to Broca's area called the arcuate fasciculi. And they communicate with each other. So you're interpreting the language that you hear, and then you're speaking the language you're replying in, and that's where the motor movements come from for your tongue and your pharynx and things like that. Wernicke's area and Broca's area. Wernicke's area is called also known as the general interpretive area. All right. Brodmann's areas. The brain is mapped. I don't want you to memorize Brodmann's areas at all. Please do not. There's over 50 of them, or about 50 of them, someplace in that area. And basically, I want you to know that we've mapped the brain. And by the way, not everyone is exactly the same. You can see that it looks like 39 and 40, and I don't know how much of each would be about the Wernicke's area. Well, I mean, this is mapped for an average human being. Someone else's Wernicke area could be slightly different than that. Wernicke's area is typically in the left lobe. It's almost always in the left lobe. And by the way, the Broca's area, I'm sorry, the left hemisphere. So Wernicke's area and Broca's area are almost always in the left hemisphere. That's not an absolute rule, but it's almost always true. So the brain is mapped. And this is just a table showing you what the different lobes do for what primary cortex they offer to you. And you should know these. You should memorize this table. We do have hemispheric lateralization, and that means we have a left hemisphere and a right hemisphere, and they divvy up the jobs between them. For example, you can see that Wernicke's area and Broca's area it's almost always in the left hemisphere. It's rarely in the right hemisphere. What the left hemisphere does reading, writing, and arithmetic. So it does writing, it does arithmetic, and the Wernicke's area, by the way, would help do reading because you're actually reading in a certain language. So reading, writing, and arithmetic is the left hemisphere. So what does the right hemisphere do for you? It does spatial analysis. So you can see right here, spatial visualization and analysis. It's your artistic lobe or artistic hemisphere. Your right hemisphere is your artistic hemisphere, and of course these two hemispheres talk with each other through these commissural fibers like the corpus callosum and the anterior commissure and the posterior commissure. So there is hemispheric lateralization. When hemispheric lateralization is lesioned and the two hemispheres aren't talking to each other, say surgically or in a disease state, then you do see a ramification of that, but then your brain does adapt and take over for the lateralization that once existed. So what I'm saying is some of these jobs in the left hemisphere can be picked up by the right hemisphere and vice versa. All right, electroencephalograms. These are picking up brain waves. So you hook these monitors to your scalp, and you can see them hooking the monitors to the scalp right here, and they're picking up brain waves. So what kind of brain waves do we pick up? Well, the first kind of brain wave we see is in an awake, or actually maybe starting to doze adult, a resting adult, we say. but you actually might be falling asleep and still have alpha waves, but you're not in any kind of deep sleep at all. So a quiet resting adult will have these alpha waves. You can see these alpha waves right here. Now, if you're concentrating, you're doing your calculus, you're studying for biology, you're going to get these beta waves. Beta waves are when you're intensely concentrating. And you can see the difference between alphas and betas right there. Theta waves down here, these are largely abnormal in adults. Sometimes adults will get them if the adult is intensely frustrated, they might get some theta waves. But these are largely abnormal in adults, but they're quite common in children. So you should only see theta waves in kids, or if you see them in adults, it's usually due to an intense frustration. And then delta waves are seen in sleep. This is a sleep wave right here, these delta waves, and it's typically a deep sleep because, as I mentioned earlier, the alpha waves can be seen as you start to doze off. And we'll talk about sleep in a few minutes. So you can see the delta waves there. So these are EEGs. Here's the coolest thing going on these days. Coolest thing going on is we have mechanical devices that you can calibrate to operate from a given brainwave. And Jerry Zoinetti did this at Super Science Saturday. And what happens is, and last year what he did is you raised a ball in a tube by concentrating intense concentration. So the instrument was calibrated to beta waves. And this is going to be pretty cool because think about what we can do. You got people that lose limbs and they have a prosthetic device on there. Well, you could put an articulated prosthetic device on there that's calibrated to your brain waves, and you could actually make that articulated prosthetic device move. And if you got good enough at it, and the calibration was good enough, and the equipment was good enough, you could have a prosthetic limb that moved just as well as one of your own limbs. And that's where the science is going with this. Lastly, our brain does have a synchronization. We have a pacemaker mechanism in our brain, and the electrical activity in our brain within a hemisphere and between hemispheres is synchronized. And we do have damage to the brain where it gets desynchronized, and desynchronization usually leads to seizures. And there's lots of different kinds of seizures out there. There's petit mal and grand mal seizures, etc. And there's drugs that help to treat these seizures. But it's usually due to a desynchronization in your brain. And it could be a temporary or a permanent desynchronization, as you've probably heard of seizure disorders. Sometimes they're mild and temporary and go away, and sometimes a person will have recurring seizures their entire life and have to be treated pharmaceutically for them. All right.